coming in. All right, I think I will get us started. Let's see, hold on one moment. Yeah. So welcome everybody to Neurology Grand Rounds. I'm Jordan Smoller, actually from the Department of Psychiatry, but I'm also the director of the Omics unit of the Mass General Division of Clinical Research. And this Grand Rounds is one of our series of Grand Rounds that's sponsored by the DCR in Genetics and Genomics. We're very grateful to the Department of Neurology for providing this opportunity. I just want to, before introducing the speaker, give a plug for our Omics unit. We're available to provide free consultation to MGH investigators who are interested in designing or conducting omics-related research. So if you need any help with study design or identifying collaborators or core facilities or anything else omics-related, please reach out to us through the DCR website, and I will be happy to connect with you. And now it is my distinct pleasure to introduce today's Grand Round speaker, Dr. David Goldstein. Dr. Goldstein is a pioneering and world-renowned human geneticist whose work has focused on human genetic diversity, the genetics of disease, and pharmacogenetics. And he has recently been serving as a director of the Institute for Genomic Medicine, and he is professor of genetics and development at Columbia. And the mission of that work is to integrate genetics and genomics into research, patient care, and education. He previously directed Duke University's Center for Human Genome Variation, and among his many accomplishments, discovered a series of disease-causing genes and syndromes, especially in the areas of neurology and infectious disease. He has served as advisor to numerous pharmaceutical companies, including as AstraZeneca's chief genomics advisor, where he led an initiative focused on the discovery of new targets and biomarkers linked to molecular mechanisms of disease across multiple therapy areas. And more recently, he co-founded Praxis Precision Medicines, which is a public clinical stage biopharmaceutical company that's translating genetic insights into developing therapies for CNS disorders. He's currently the CEO of Actio Biosciences, which is another company focused on using bioinformatics to systematically interrogate the genome and identify novel disease targets and treatments for Mendelian genetic diseases. And I have had the privilege of working with Dr. Goldstein in his role as a PI in the NIH All of Us research program, which, as you may know, is the largest ever cohort study in biomedical research. And I'm just thrilled that he agreed to talk to us today about the role of precision medicine in drug discovery. So please join me in welcoming David, and I'm going to turn it over to you, David.
Jordan, thanks so much.
Actually, before you start, if folks have questions, please put them in the Q&A, and we'll try to get to them after the talk. Thanks.
Jordan, thanks so much for that really kind introduction. And let me say it's a particular pleasure to be addressing both the psychiatry and neurology communities. I've thought for some time that we should really be thinking about genetics as a starting point in similar ways in both psychiatry and neurology. And it's also a pleasure really to be invited by Jordan, someone whose work I have really very long admired and who was very much a kindred spirit while I was working with him in the All of Us initiative. What I'd like to do is try to give you a sense of how we're viewing precision medicine today, both in terms of its relevance to the delivery of healthcare, but also in terms of its relevance in drug development. Let me just say, as Jordan noted, that I am involved in two companies that are precision medicine focused. So I want to make that disclosure. Of course, some of the scientific perspectives that inform the approaches in the companies will be discussed, but I will not be plugging any products or planned products or anything like that for either of the companies. I won't be talking about any marketed or planned to be marketed products. What I will be talking about really is how I've been thinking about approaching stratified medicine, really both from the perspective of the use of genomics in the clinic and the perspective of drug discovery. And I want to really start with my own characterization of what we're trying to do in stratified medicine or precision medicine. The fundamental goal, in my view, is to use omics approaches usually, but whatever approach that can help you to differentiate amongst patients. Use these approaches to identify subgroups of patients that have mechanistically similar diseases. And this is something that that clinical classification often fails to do. In some cases, fails to do very spectacularly. WES is one important tool in this, arguably right now, one of the very most important tools. But of course, there are other omic approaches, and it was particularly nice to hear Jordan describe their approach for helping investigators to plan omic studies. In order to use sequencing or other omic approaches effectively, it is really absolutely essential to deploy these diagnostic tools very, very broadly. And that's really one theme that I want to emphasize with some of the examples, that the selective application of these diagnostic approaches has given us really a very biased view of ways in which patients can be subdivided. And it really is only by taking a much more comprehensive approach to the deployment of these tools that we will get a less a biased approach. That, of course, does mean if we take a more comprehensive approach that we will, for example, end up with patients where after performing, for example, whole exome sequencing, we don't identify diagnostic variants. But I would really suggest that that is absolutely fine. These tests are not very expensive. And because of how often we bump into diagnoses in areas we weren't expecting, or diagnoses of a sort we weren't expecting, it really does argue for a comprehensive approach. So this is the typical paradigm where you start with a patient that has a condition that is not mechanistically explained, even if there is a clinical diagnosis, and deploy some kind of omic approach to better understand the condition. For example, whole exome sequencing of an affected child and his or her parents, and one then sifts through these data to try to find a genotype that is responsible. I will emphasize here that I am focused entirely on the identification of variants that are thought to have a major impact on disease, either individually causal or that have a very strong influence on risk of one or more diseases, as opposed to use of a large number of variants that have more modest effects on risk through, for example, polygenic risk scores or something like that. That's not because I think that polygenic risk scores are of no utility. It's just not where I focus. So this is a typical paradigm, and then one tries to look through to find these responsible variants. And if you look through a range of different therapeutic areas, what you find is that the diagnostic yield from these approaches, either whole exome or whole genome, is really very, very high. And this is something that I think really the community should be very, very proud of, having made such a valuable contribution to clinical diagnostics. And I would also invite people to think about just how big a challenge this really is and how well it's been accomplished. The genome is, as we all know, a big, complicated place, and there is lots and lots of functional genetic variation in every single genome that you look at. And yet, we can routinely today start with a patient with an unexplained condition with no idea whatsoever about whether there's a responsible single genotype, or if there is, where it might be. And we can look through the almost entirety of the coding variation in that individual, and and pluck out a single genotype that is responsible for disease presentation. And this really is just really a truly remarkable a contribution to genomics. I will also say that it is clear that the vast majority of these declared diagnoses, either from a research group like ours or ones that are at Harvard, or from diagnostic labs, the vast majority of these declared diagnoses are accurate diagnoses. There are a whole bunch of different analyses that you can perform that show really very clearly that the vast majority of these diagnoses are accurate. So across a broad range of therapeutic areas, upon sequencing, you might end up with a quarter or 50% or even a little bit more of patients that are explained through the genetic analysis. And this then allows you to divide the patients into groups that have distinct underlying mechanistic causes of disease, with implications for prognosis, with implications for the best treatment, and so on. We at Columbia, at the Institute, which I directed for the first 5.5 years, and Ali Gharavi now directs, sequenced a little over 10,000 patients with a broad range of different presentations. And I want to just give you a little bit of a sense of the kinds of things that we've seen. And we really did consider the application of genomics across really the full human lifespan. And we feel that this was really vital to getting a sense of the kinds of differences that we see in underlying genetic causes dependent on the presentation. And this really did begin right in the beginning with efforts focused on prenatal presentations, both stillbirths and products of conception, all the way through to very late onset presentations, adult forms of CKD, ALS, and some neurological presentations that actually appeared in the 60s and 70s for some individuals with positive diagnoses. And looking across this very broad range of presentations, we're really able to show some pretty sharp differences. And I won't walk through all of them that we've seen, but one that I found really striking is that if you look at the adult presentations that we had characterized genetically, and you ask in what genes you see variants that carry bioinformatic signatures of being pathogenic, what you see is that the vast majority of those variants are found in genes that are already known to cause one or more strong familial diseases. And my interpretation of that pattern is that for postnatal Mendelian strongly familial diseases, we actually now know about the vast majority of genes. And I think that's actually quite consistent with how novel gene discovery for familial diseases is, in fact, I think pretty clearly asymptotic. And I can say from my own experience that when diagnostic sequencing was first being rolled out, we were bumping into completely novel genes that hadn't, novel in the sense that hadn't been connected to any familial disease yet. We were bumping into them very regularly. And then in the last, say, 2 or 3 years, that happened less and less frequently. Not that there aren't any new ones coming out, but it's just less and less frequent. And I think that's because we've actually got most of them. Now, some people don't think that that's true, and they think that the vast majority of genes in the genome will have some strongly familial presentation. But for a presentation that is important enough to cause people to have to go to the clinic for conditions that geneticists think are a Mendelian disease, I think we actually have most of them. And I think what we don't yet know is actually the full phenotypic consequences of mutations in the genes we already know about. And what is still happening regularly is that we are expanding the phenotype associated with a number of diseases. And that's actually extremely challenging to do. We've actually gotten really pretty good yet thinking about the statistics required to say this gene is a Mendelian disease gene. Not every single one that's thought of is really a real disease-causing gene, but the vast majority of them are. But we actually are not as good in terms of a statistical framework for saying, oh, I now know that this gene is associated with this novel phenotype that I didn't know was associated with before. I think that's where a lot of the action is now, trying to really understand the consequences of variation in the genes that do cause familial diseases. And I will plug the All of Us research program here, which I think investigating those kinds of phenotypic consequences of variation in disease genes is something that it could actually end up being quite good at doing. But the pattern that I just described of the variants of bioinformatic signatures falling in these known genes for postnatal conditions is in marked contrast to what we see in the prenatal conditions. There, we saw a very clear elevation in, for example, stillbirths of variants that are in genes that are the very most intolerant genes in the genome. And in fact, this addressed a longstanding puzzle in the community of investigators working on so-called intolerance scores, where we had noted for a long time that disease-causing genes tend to be intolerant to functional genetic variation, standing variation in the human population. But there was a set of the very most intolerant genes, for example, that had virtually no loss of function variants anywhere in the human population, that were not implicated in disease. And so we often wondered what was happening with these. Well, it turns out that loss of function variants that we see in stillbirths are very enriched for these very most intolerant genes in the human genome that are not yet implicated in disease. And so this is suggesting actually that there are a set of genes when mutations occur that are not compatible with development to term, and that we actually won't learn about by studying postnatal conditions. This just gives you a little bit of a sense of how we've been doing the work, the diagnostic sequencing work at Columbia. We established research protocols that governed the work. We invited patients directly from clinics to participate in sequencing studies. We centralized in one place all of the data generation and the bioinformatic interpretation. And it is this centralization that really helped us to make comparisons across groups. And let me just say that I think this is a really important thing for academic medical centers to do. I think there's a world of difference between getting a PDF report from a diagnostic company, even though the diagnoses are secure. As I said, there's a world of difference between getting a PDF where what you have is whatever the lab comments on, and actually having the underlying data really facilitating novel discovery. And then we had an analysis infrastructure that performed all of the diagnostic work. And then critically, whenever the research team felt that a variant or genotype was likely to be contributing to disease, we then interrogated the variant in a clinically certified lab, and then returned the information back. And all of this activity was supported with institutional resource. I think that last part, that the work was supported with institutional resource, is actually a really important reason that the individuals that participated in these research studies performing diagnostic sequencing are fully reflective of the diversity of the patient population that the medical center serves. And I think that this actually illustrates that the longstanding problem that genomics has had with diversity, which has been commented on now for well over a decade, can be partially, maybe even largely overcome by removing financial barriers to participation. I don't mean to imply that those are the only barriers. We actually worked very, very hard to ensure that we had inclusive enrollment in these research studies. But I do think, taking, for example, the question of whether you can get insurance to pay for a sequence or not, really can help with ensuring inclusivity in these kinds of studies. So I think this is a really important message for us to keep in mind, that it is all important that these kinds of studies are performed inclusively. They emphatically have not been, and I'll just note that more than 10 years ago, we summarized the application of genome-wide association approaches and found that individuals of European ancestry were included in such studies at a rate of about 9 for every individual of non-European ancestry, and that extreme imbalance in geographic ancestry participation has persisted through and has papers lamenting it written in high-profile places every couple of years without it really being overcome. The All of Us research program is very much focused on correcting these imbalances and is doing a good job of it. And I think that when you perform diagnostic sequencing clinically, making an effort to be inclusive, but also removing financial barriers, then you can really end up with a diverse cohort. It's also really quite exciting to see that when you perform this kind of sequencing comprehensively, you end up with a very large number of diagnoses in individual genes at a single academic medical center. And so this is the most common diagnoses are summarized here. And the real point here is that I can tell you from a number of conversations that I've had with pharmaceutical companies that there's a lot of concern about the challenges of performing genetically stratified trials. And the concern is quite straightforward. If you have a relatively rare condition, say, and you not only have to find patients with that condition or particular clinical presentation of it, but you further then have to find patients that have a particular contributing genotype, then the feeling is, well, maybe we're going to end up with one patient from each academic medical center, and to run a trial even with 50 patients, we're going to have 50 academic medical centers as sites, and it just is massively cumbersome. This summary indicates that things probably are not as challenging as that. And as long as you have cohorts of patients that have been carefully characterized genetically, you end up with many patients with disease due to mutations in an individual gene at just one academic medical center. And so this suggests to me that a stratified approach to trials, genetically stratified approach, is in fact quite feasible, as long as we have a number of academic medical centers that have fully genetically characterized patients, and that the genetic characterization has been carefully adjudicated, and that you have these genetic data available alongside clinical data for patient selection. I would actually call this out as a major priority for the community if we actually believe in the potential of stratified medicine. And I'm going to come back at the end to really say we have very good reason to believe in the potential. And to the extent that we all think that's true, it really is the priority to get these data, these genetic data, in shape to support stratified medicine. And that really means that the data have to be accessible for patient selection for trials. And I would really call that out as a high priority. I'd also like to emphasize, especially since Jordan invited me to the talk, I'd like to emphasize that these strongly acting genetic risk factors are absolutely not restricted to some of the disease presentations that we have come to expect them to be found in. So it is now, I think, very well appreciated that if you have a severe epilepsy presentation early in life that is not explained by something symptomatic like TBI, that you really need to do genetics, because there's a very high diagnostic yield, maybe for some of the more severe presentations, maybe as high as 60 or 70% of the children that get sequenced have a clear genetic diagnosis. And schizophrenia had long been thought of as being much more dependent on common variant influences of more modest effect. Harvard and others led a very exciting study fairly recently called Schema, showing that you do end up with genome-wide significant evidence of rarer, higher impact risk factors upon sequencing very large numbers of patients with schizophrenia. But Anthony Zogby and the group at Columbia had the view that the impression that one had of schizophrenia genetics may have been somewhat biased by a tendency to include less severe schizophrenia patients. And of course, we're well aware in neurology, in fact, just as I was just outlining for epilepsy, that the genetic architecture is dependent on the severity of the presentation. And so he designed an approach to really focus in on the very most severe schizophrenia patients in terms of being refractory to treatment and in terms of having a presentation severe enough to warrant residential care. And in a really laborious effort, partnered with a number of residential care hospitals in New York in order to appropriately enroll patients that had been long-term residents. And then he performed diagnostic sequencing on really a relatively small number of individuals that on the basis of these criteria were the most severe. And strikingly, even in a quite small cohort, he saw a very strong elevation of rare high impact variants. And in fact, he saw an elevation of these kinds of variants that was well beyond what you saw in what he referred to as typical schizophrenia, here meaning really just schizophrenia not collected purposefully, as I described, to be the most severe, well in excess of what you see in typical schizophrenia. And in fact, with high error bars, but along the lines of what you see in some neurological presentations. And I think this is a really important emphasis that the exact presentations that you include really have an important impact on the role of these higher impact variants. And so even thinking about stratified medicine focused on underlying responsible genotypes has application even in disease areas that had often been thought to be much less dependent on those kinds of high impact variants. So what's the point of all of this? The point is quite clearly to identify genetic drivers of disease that can help us to more effectively treat disease. And it certainly is the case that sometimes when you get a clear genetic diagnosis using genetics, that it has a transformational impact on the care of patients. We had a really moving example of this in our very early diagnostic work when we sequenced, along with Vandana Shashi and others at Duke, Kara Green, pictured here, of course, with full permissions. She presented at the Children's Hospital at Duke with a progressive neurological disease that was one of these typical situations for diagnostic sequencing where the clinical team really had no idea what it was, but they knew it was very bad. They thought it might be an autoimmune condition. When we sequenced Kara and her parents, we found, in fact, that she has this very rare and completely treatable disease called Brown-Vialetto-Van Laere syndrome. And the disease is characterized with a number of features I won't go into, but including extreme upper body weakness. And early in her life, whenever Kara fell over, she fell flat on her face because she couldn't raise up her arms. But it turns out that this disease is due to knockout mutations of the vitamin transporter. And the treatment is to give patients a lot of the vitamin. That treatment was initiated for Kara within like 36 hours of our seeing the genetics results. And then she virtually immediately moved into a really, really improved developmental trajectory, and within a number of months began to have a near normal developmental trajectory. And that led to Kara's parents regularly sending the clinical team photos of her happily lifting her arms. And this is rightly a precision medicine feel-good story, because it is in fact exactly what we're trying to do in precision medicine. Go from not knowing the underlying cause of disease to knowing the underlying cause of disease, and then deploying a highly effective treatment for exactly that kind of disease that would be of no use outside of that context. So that really is a precision medicine therapeutic, really is something for us to feel good about. And it really is exceptional. And I really want to emphasize that some of the messages that are coming out that diagnostic sequencing normally informs care, I think are very, very misleading. So there have been a number of papers that, even if they're written a little bit carefully, have often been construed as showing that, for example, the majority of time you get a diagnosis for a patient with diagnostic sequencing, it has an impact on care. I don't think that these assessments are technically inaccurate, but I do think that they're misleading, because I think it's really important that we differentiate between a transformational impact on care, like the Kara Green example shows, and a very modest impact on care. And by emphasizing that genomics is so important that we normally get an impact on care, I think we kind of miss the point that what we're really trying to do in knowing the underlying cause is find a highly effective treatment. So to give you an example of exactly what I mean from neurology, I think that it is not really tremendously helpful to, for example, find an activating mutation in a sodium channel gene, SCN8A, for example, and then say that genomics influenced care because it indicated sodium channel blockade as a potential treatment for that patient. I think most epileptologists will confirm that if you have a patient that has uncontrolled seizures, and you don't think it's Dravet syndrome, that they're going to end up getting a sodium channel blocker sooner than later. And so you can't really say that you've transformed the care by having a genetic result that maybe, or maybe even not, change the order somewhat of when you might try a sodium channel blocker. I'm not saying that that's useless. I'm saying it's not a transformation in the care of the patient. So if you think about where we can really make a truly big difference and deploy highly effective treatments that would not otherwise have been used and that really make a difference, if you look at it from that perspective, which I think we should, so we can really tell how well we're really doing with what we want to do, then it becomes clear that these kinds of examples are spectacularly rare. So, we have sequenced many thousands of patients, and we have a handful of these. So you can actually look at direct experience to show that it doesn't happen that frequently. But you can also think about, from first principles, what we're really doing when we do this kind of diagnostic work. And I think that's really important to do. What we're doing is we're starting with the patient with an unexplained condition, and then we're looking at their genome, and we're actually linking that patient up to an already known Mendelian disease gene, which you can go and read about in OMIM or whatever. So we link up the patient up to one of the 4,000 plus already known Mendelian disease genes. And sometimes there are many phenotypic presentations for one of those genes, but we link a patient to one of those genes. And if you start then with the gene list and say how many of these genes that cause these strongly familial diseases have highly effective treatments like Brown-Vialetto-Van Laere syndrome, the number is very, very small. So it really shows us that what we really need to do if we want to expand the domain of application of precision medicine is really get much, much better at development of effective treatments for these Mendelian disease genes. And the majority of them are actually genes that cause quite rare diseases. And this has been a major barrier to drug development. There's been a lot of talk about repurposing existing medicines for rare diseases. And I can tell you that, of course, we should check, but there's a world of difference between knowing the underlying cause and saying, I'm going to take one of the only several hundred approved medicines, or a little bit more than that, and see whichever out of that very small set of compounds is the best for this genetic disease. That's very, very different from starting with the disease presentation and actually running a drug discovery effort to find the most effective treatment for that disease. And the problem is that the rarity of the presentations makes the drug development effort unattractive, even if for the particular genetic disease, it would actually be a relatively straightforward drug development effort. So now here's an example of where some of the perspectives of the companies I'm involved in comes in. So I want to make that disclaimer. But I think there is an economic argument to be made, even beyond having effective treatments for the very rare presentations. If you actually look at the medicines that have been approved, and then you ask what they target, it turns out that approved medicines very, very preferentially target genes that cause Mendelian disease. And this is an illustration here on the graph that we did a few years ago with Joe Hoystick in the group and others, just showing that the primary targets of approved medicines used for common indications are very preferentially Mendelian disease genes. I think the reason for this is very clear. These Mendelian disease genes cause Mendelian disease because they are involved in very important areas of human biology, and then modulation of those ends up being relevant outside of the rare disease. And the importance here, I think, is that if we as a community decided that we were going to systematically start pursuing effective treatments for these devastating rare diseases, we would not only end up with a number of effective treatments for very rare diseases, but we would also, in fact, not almost certainly, based on these kinds of assessments, we would certainly end up with modulators that can be used more broadly too. So I think this makes an economic case. I think it also makes a medical case that this is just a good path to having important modulators. And I think the way to view these Mendelian disease genes is that they sit in critical areas of human biology and kind of represent choke points for a broad range of different disease presentations, rare and severe, and more common too. So this, I think, really is my pitch, that it would make a whole lot of sense for us as a community to work much harder on treatments for the rare diseases. I will say that it's not as if rare disease therapeutics is not getting attention. It's getting lots and lots and lots of attention, but it's the more common rare diseases that are getting attention, and those are relatively few, because there are enough. Cystic fibrosis is the paradigmatic example. There are plenty of patients to make the economic case for cystic fibrosis alone, but many, many, many of the severe dominant disease have very few patients. And my argument here is that they still represent very important areas of focus for drug development for these reasons. Okay, so all of this has got us thinking about where there are opportunities for drug development. I am not going to talk about the examples that we're working on in companies. It's an academic talk. So I'm going to talk about an area that is too hard right now for any of the companies. And that's trying to think about the idea of transcriptomic restoration in neuropsychiatric diseases. This is something I'm actually really, really excited about, but though I think it's very challenging. Recognizing that so many of the genes that cause disease, neuropsychiatric diseases, have a common underlying mechanism for causing disease. We started thinking about whether we could take a similar approach for a large set of genes, so it doesn't have to be a bespoke effort for each individual gene. And when we were looking at genes from this perspective, we quickly realized that genes that cause disease apparently through the dysregulation of large numbers of genes represents one of the very most important underlying mechanistic classes of disease causation. And so we summarized, Ryan Hinza and others in the group summarized the genes that cause multiple neuropsychiatric diseases by modifying the expression of lots of genes, for example, transcription factors and chromatin modifiers and RNA binding proteins. And we found that almost half of the genes implicated, for example, in autism are in that sense regulatory in nature. Maybe a quarter of the epilepsy genes, and out of the schema results, 20% of the schizophrenia genes that we know about so far. And so this got us thinking that probably in these cases, what's happening is that you've got, for example, an RNA binding protein that has mutations in it, so it's not serving its appropriate function and helping to have pre-mRNAs processed into mRNAs. And so you have lots and lots of genes that end up misregulated because of that. We started thinking that what you could do is identify the genes that are misregulated due to these mutations, and then identify compounds that act on, for example, key regulatory points in order to restore the normal expression of as many of those genes as possible. And that might be a therapeutic strategy that could be consistently deployed for these classes of genes. So in work that Andrew Ressler and the group did, we built organoid models of one of these presentations, HNRNPU deficiency, on behalf of a patient family. And we carefully characterized the transcriptomic dysregulation that these mutations cause. And we actually found really quite encouraging results for this approach, but also somewhat daunting results for this approach. On the encouraging side, what we found is that if you compare the genes that are dysregulated in the organoid model based on human cells to the genes that are dysregulated in a mouse model, you actually get a high correlation of dysregulation. So that suggests that we are able in these model systems to identify genes that are dysregulated with a consistent enough pattern across species, across models, that they probably are representative of what's happening in vivo. So if we can find restorative agents in these models, then perhaps they would also be useful in vivo. On the more daunting side, we also showed that the dysregulation varies dramatically, not only by tissue type using single cell sequencing, but also by developmental stage. So we found that the organoid dysregulation correlates well with embryonic mouse dysregulation, but not nearly so well with postnatal. And we found really dramatic changes in what genes are dysregulated pre and postnatally in the mouse. This is a real challenge for this paradigm, of course, because it tells us that we don't really know where and when to characterize the dysregulation of gene expression in order to try to correct it. I think this really just illustrates that this is very hard work to do, but I do think it also shows that it is work that can be done. And what I think is exciting about the opportunity is that if we are able to work out paradigms that are effective, it will apply to a very, very large number of these disease-causing genes. So I want to wrap up by just emphasizing that the really spectacular growth of genetic data sets really do help us to think about taking more systematic approaches to drug discovery for rare disease genes. And I just outlined an approach that applies to a set that operate through dysregulation of lots of genes. But if you use simply databases of identified pathogenic and likely pathogenic mutations, those are actually sufficient to discriminate to a degree between genes that cause disease due to a gain of function and that cause disease due to a loss of function. And this is important because, of course, we need to know if we want to modulate the protein in a rare disease, we need to know what direction we need to modulate. And it's really striking that up until recently, there are actually a lot of disease genes that we could be confident cause disease, but there were so few mutations out there and no convincing functional biology that we didn't actually know what direction of effect the mutations caused in patients. And so we wouldn't actually know what to do to modulate the protein. But now there's been so much diagnostic sequencing that there are very large numbers of genes that don't have convincing functional biology to show you what the mutations do, but that have, for example, a sufficient proportion of identified pathogenic mutations that are missense versus loss of function, that you can be confident, for example, the gene is not a haploinsufficient disease gene. If the gene is haploinsufficient, so you get disease because you have only one working copy, then you always see a relatively large proportion of loss of function mutations. Missense can cause a loss of function too, of course, but there are many frame shifts, stops, and so on. And those accumulate in the database because they're seen often in patients. If it's a haploinsufficient disease gene, that does not happen if it's a gain of function where you see always an excess of missense mutations. So this is really just to emphasize that we have these growing data sets that really allow us to understand much better, even without the hard functional biology being done, what direction we need to modulate. And this allows us to start thinking about systematically dividing up the genes that cause rare diseases for different kinds of drug discovery efforts. And I won't walk through the kinds of drug discovery efforts, but just would emphasize that some of them are really, really straightforward, where it's perfectly clear that the mutations are, for example, gain of function mutations in a protein that is very clearly modulatable with small molecules. And there are a lot, and just kind of a plug for us to call to arms for us to really get to work. There are a lot of genes like that that cause devastating presentations, where if you looked at it from the drug development perspective, you would say this is a tractable proposition. That's something that I think we need to address sooner than later, because those patients could have an effective treatment if we were to launch appropriate drug discovery efforts. And now finally, just to kind of address some of the key challenges to precision medicine that have been raised. I addressed a couple already. I think the clinical trials where they're genetically stratified may be a little bit more feasible than it has been considered. I think the economic case may be better than has been realized because of the potential of broader application of modulators of rare disease genes. And I want to end by addressing the idea that these kinds of approaches are only applicable to a very small proportion of patients that have these strongly acting mutations. I've already hinted that they appear in more places than expected, but I now want to address that a little bit more directly. So in work in collaboration with Ali Gharavi, actually in work led by Ali Gharavi in nephrology at Columbia, we looked in collaboration with AstraZeneca at the diagnostic yield of adult onset kidney disease. And really strikingly, we found that 10% of individuals with adult forms of chronic kidney disease have an absolutely clear Mendelian cause of their diseases. And moreover, in many of these cases, the Mendelian form, even often very well-known forms, was not clinically recognized. Now, if you keep in mind that chronic kidney disease is one of the very most common of the common diseases, and clearly highly complex, so a classic archetypical common complex disease, it's really quite remarkable to show that that 1 in 10 have a genotype that is responsible or largely responsible for disease presentation, meaning that we now have the tools to subdivide this very important common complex disease into groups that are mechanistically distinct, and many of the underlying genetic causes are very, very different from one another. And one would in fact strongly suspect that the optimal treatment for patients would be dependent on which of these very distinct underlying causes of disease an individual patient has. And we of course already know that's true with many examples. For example, an individual that's diagnosed with Alport syndrome, you would not put onto immunosuppressive therapy. And that provides a direct illustration of clinical utility, where we actually showed that not only were many of these patients diagnosed with Alport syndrome, but actually it was often clinically not recognized. And then having performed the genetics, you would know, for example, not to put those patients onto immunosuppressive therapy. So this, I think, really does show the importance of a very broad application of these diagnostic sequencing approaches, but it also shows the importance that we run trials that take account of this underlying heterogeneity. And it really does worry me that we continue in so many cases to try to identify medicines that work across the board for something like CKD, or for example, something like refractory epilepsy, when we know now with certainty that those patient populations are incredibly heterogeneous. And we're really asking a medicine to do something that's very difficult to do, where we simply take everybody independent of underlying cause and then hope that a medicine works well enough across the board, when we know that, for example, refractory epilepsy, there will be tens, perhaps many more, distinct underlying mechanistic causes of disease represented in one single trial population, and also for CKD. To me, it's very, very clear that the answer here is that we need to take account of that heterogeneity. Exactly how to take account of it is something to be worked out. I would say a starting point is actually to characterize the underlying heterogeneity and ask how it correlates with response. That creates challenges for clinical trial design, admittedly, but I would say that developing the appropriate framework for taking account of this heterogeneity is critically important. And then just finally, to conclude, I don't know what others' views had been for OCD, but I myself had taken a view that this was in fact one of those disease presentations that were largely dependent on common variants of modest effect, as opposed to rare variants of higher impact that might represent good focal points to get into disease biology. And up until recently, there actually hadn't been genome-wide significant evidence implicating rare variation in OCD. We recently performed sequencing studies in collaboration with Jerry Nestet and others in OCD, and we found really quite clear genome-wide significant evidence of rare variants in the SLIT-RK5 gene involved in synapse formation. This is a gene actually that has long been a focus in OCD. Genetic studies had implicated it, not with genome-wide significant evidence. Animal model studies had implicated it, and as I think those who know me will know well, I have always been ultra suspicious of genes that have good stories, even animal models associated with them, in particular in neuropsychiatry, but without genome-wide significant evidence for the genetics itself. And usually those suspicions are well-founded, I think, but in this case they weren't. SLIT-RK5 is certainly really implicated, and I think it's a very exciting opportunity, and emphasizes that that these high impact variants that do, I think, represent good focal points for developing treatments really do show up in a very, very broad range of presentations. And there I will stop, and just to really emphasize and come back to this call to arms, that I really do believe, having thought about it from this perspective for a while, that many Mendelian disease genes represent terrific opportunities for drug development, and that we could end up after those efforts with treatments not only for a rare disease, but also for more common ones too. And this is something that I strongly believe we should be working on together, both in in the private world and on the academic side, which is responsible for my jumping back and forth between the two. So thank you so much for your attention. Really has been a pleasure and an opportunity to discuss these things with you.
Thank you so much, David. That was fantastic. And we have about 2 minutes left, but we have some great questions. So I may ask you to try to answer these quickly.
Okay.
I'm looking at a couple right now. One from Kevin Staley, amazing talk as always. Most epilepsy drugs are effective at high concentrations that are unlikely to be affecting a single target. Target identification has not been wildly successful so far in epilepsy, e.g. quinidine for CKNT1 mutations. Do you think that for some conditions, multiple targets may need to be engaged for clinical efficacy?
That's a really great point, actually. So I completely agree, and it's interesting that sometimes when you get more selective, the drugs don't work as well. So yes, I wonder if that's a function of how the anti-epileptic drugs have been discovered, and you could imagine that if we were to focus on more mechanistically distinct subgroups of epilepsy patients, that would be less the case. But that said, that certainly is the way it works now. I think it's an interesting point, and maybe it does suggest that in some cases we should be thinking about engagement of multiple targets.
Another question from Andy Cole. Single gene mutations are particularly common in epilepsy, for example, and particularly tractable in therapeutic development. But in many disease states, sequencing efforts lead to polygenic risk scores. While these are typically thought of as markers or risk factors rather than specific causal elements, how do you think about converting genetic information into therapies in truly polygenic disorders?
To be honest, and I don't want to go back to, some of us are old enough to remember what had been called the GWAS wars. I don't want to go back to those at all. But it's just not the area that I focus on. I will say that I think that it's clear that there are ways that you can use polygenic risk scores that are clinically meaningful. There's really no ambiguity about that. I'll just give one illustration that it's very clear to me that that using polygenic risk scores in clinical trial populations can be very valuable, because you could easily select for patients that have a different event rate. I do think that there's a really big difference, though, between being able to identify a protein in a pathway that individually can have a massive impact on disease as a therapeutic target, as opposed to modest changes in expression, say, of tens or hundreds of genes, which I think do not actually represent starting points for drug development in my view. And I would say that's particularly true because not only are these polygenic risk scores diffuse, modest impact individually, but as far as can be discerned, they don't actually load up much in individual pathways. If they loaded up in individual pathways for specific diseases, I'd say that it highlights the pathway, but by and large, that doesn't happen. So I think for the kind of therapeutic efforts I'm talking about, I don't think that they are informative.
Perfect. Okay. Well, we didn't get to every question. I apologize, but that was really terrific. And we are at the end of the hour. David, I want to thank you again for a great talk. Thanks again to the Department of Neurology for hosting this Genetics and Genomics series. Everybody have a great rest of the day. Thanks.
Thank you. And any other questions people want to ask, please just shoot me an email. Thanks very much, Jordan.